Welcome to the 783rd monthly meeting of EPAASM. This is also our first meeting in 2021, so Happy New Year. Today we have a very special guest, Dr. John McLanos from Harvard Medical School. Before we go on, I just want to refresh your memory on the Zoom etiquette. So we're going to hold all the questions toward the end. So if you want to ask questions, we encourage you to hit the participant tab. And you can raise this little blue hand, and then I'll unmute you so you can ask your questions. Alternatively, you can enter your questions in the chat box, and I can relay those questions. As a service, this is like a test drive for NPR. So please join ASM. We provide a community, as well as a lot of resources, as well as career development. And there are a lot of micro online resources that you can watch, either sessions or posters you can look at. And we also encourage you to join the local chapter EPAASM when you do. So today I have the distinct honor to introduce Dr. John McLanos. He's one of the few people that really don't need a lot of introductions around ASM members. But I'll try. So he's the Adele Lehman Professor. He's served as the chair of the Department of Microbiology and Molecular Genetics for 20 years. He's been elected to the National Academy of Science. He's won numerous prestigious awards like the Eli Lilly Awards, the AAS Newcomb Cleveland Prize, the City of Medicine Award, the Drexel Prize in Infectious Disease. He's co-authored more than 270 articles. He's the giant in bacterial pathogenesis. His earlier work really teased out the FOPQ regulations in Salmonella. His work on TCP and cholera toxins pathogenesis, look at phage interactions with Vibrio. He's come up with the IVAT system to look at in vivo expression of virulence genes. And lately, he's been working on the discovery and identification of type 6 secretion system, that bacteria use this secretion system to interact with other bacteria for competitions. For example, how pathogens kill off microbiome to gain establish a niche. And to invite John out, I tried to plan it really well. I said, hey, John, we're some of your former trainees in the Philadelphia area, Amy from Drexel, Jay at Penn. Why don't you come out? We can have a reunion and you can see the kids. And if you didn't know, John is an avid poker player and a single malt Scotch connoisseur. So I thought I planned it out perfectly. He was going to come out on the 777th monthly meeting. And coincidentally, it's the Patrick Pickett meeting, so we would have whiskey at the meeting. Unfortunately, COVID hit, and so we had to cancel the meeting. Fortunately, John graciously accepted the invitation this year again, and to give the talk via Zoom. And because we can't let the virus win, right? So on a personal note, well, let me tell us a personal story on John. Don't worry, we can edit this out. So this is very timely because a lot of students had just had their first committee meeting. And I was nervous. It was my first committee meeting. I was nervous trying to set up, the coffee, the pastries. The projector used to weigh like 20, 25 pounds. And so I was running around like a madman and I was, nervous. And I was sweating a lot. And John had this stern look. He looked at me and said, young man, are you okay? You're sweating a lot. There's nothing more important than health. So you should really take care of yourself. And my reaction was, come on, John. I'm trying to set up to present my project to the who's who in bacterial pathogenesis. And you're talking about me joining a gym. But after that, I realized John really genuinely cared about his people. Often he would go to the mat, so to speak, for his people. I just had a chat with him this weekend and he's still the same way. And so I guess to be successful, just be brilliant, be good to your people. Well, and he also made a mean margarita. So maybe if you have those three qualities, um, sky is the limit. So without further ado, Dr. John Maclanos, and today he's going to talk about protein variants networks in SARS-CoV-2. So for me to jump from bacteria to virus, I had to do a whole postdoc. John just spent a few months and then he has a paper working on this. So
okay, well, that was an awfully nice introduction. Um, I'm trying to see if I can make the light hit me just right, but I think I'll leave it at that. You don't want to look at my face anyway. So let's see. So I'll share screen, right?
yep.
And we can get rolling here real quick. So let's see, share. So the first thing I want to do is congratulate your Eastern Pennsylvania branch for your 100th year anniversary. I actually caught a little bit of that online. I thought that was really delightful that you guys have been going strong for that long. And Victor Rita, who I think many of you know as the ASM president, said some nice words, and it just really felt like there was a very nice family atmosphere to see my trainees getting involved in this. So yes, it's true. We're working on this virus right now. You know, it's one of the greatest challenges of science in our generation, but I'm not going to take full credit for that. Bill Robbins, was a postdoctoral fellow in my laboratory, but also is still a postdoctoral fellow in my laboratory, but is now got the rank of instructor. So technically, he's a faculty member in the department as well, and trying to find some areas of independence. Uh, you know, cooped up at home during the first part of the pandemic, Bill leaned on his considerable bioinformatics and computational biology skills to look for some projects that were of interest. And he got into the idea of doing protein covariance in Vibrio first and started working in that area, but eventually thought, you know, a smaller genome might be better to look at, and we could get down to the actual residues and interactions. So he started this project. I had nothing to do with it, but I did coach him along the way as he was starting to get the first results, and we started to try to understand what was going on. But if I have time today, I'd like to try to do two versions of Bill's work. One has been published, or at least loaded on the preprint server at bioRxives. That's the one I'll start with on the virus. But if I have time, I'd like to tell you a little bit about what he's doing with covariance analysis in Vibrio cholera, which is got the unifying theme that both of them are telling us quite a bit about the evolution of pathogens. So with that, I'll move on. I hope. There. Okay, so I don't have to say too very much about these viruses. You guys all know the background. Um, just pointing out that the two earlier versions of these coronaviruses, SARS and MERS, should have given us a big wake-up call. You'd be surprised to find out how, as these died off, how quickly people stopped working on them and didn't realize that this type of virus was going to come back again with a vengeance sooner or later. But they did learn some things along the way about some of the proteins I'll be talking about today, and I think all of that is going to be informative for SARS-CoV-2 as we move forward. There's been very little actual genetics that's been done on SARS-CoV-2. It's been mostly a rush to get its genes and expressed in various vectors, and whether it be RNA-based vaccine vectors or VSV pseudotypes. So if you ask any questions about the biology of some of the proteins in the context of SARS-CoV-2, I'm not going to be able to answer them, and we're not approved to work with that virus yet in our laboratories anyway. So just a background again, coronaviruses in general can cause everything from deadly diseases to just the sniffles. The common coronaviruses on the right-hand side um, are noted as the causative agents of colds. They have been around for a long, long time. It doesn't seem like there's a lot of cross immunity to them. So one has to cross one's fingers to hope that this new COVID, the COVID-2 virus that's now causing trouble in the world in the form of the COVID-19 disease, will not take the track that the cold viruses have taken, and because of the huge amplification that's going on, learn a lot more about human adaptation and suddenly become sort of a seasonal disease that is going to be hard to immunize against. But that's part of what we try to learn in our analysis, is learn more about Achilles heels that these viruses might have that might inform vaccine development. A little bit about the virus: 30 kilobase RNA genome, largest enveloped RNA virus. More nucleotides mean more chance to mutate. Um, the virus is a single strand positive RNA virus that has 10 genes. I think it's actually closer to 11 genes, count one of these open reading frames. But anyway, it encodes about 26 proteins because there's lots of polyproteins that get proteolytically processed and generate more than one protein in the end. Um, like I said, it's an RNA virus, so I think we have to worry about fidelity of synthesis of RNA, because RNA viruses are notoriously poor at fidelity. So they potentially have the possibility of having a lot of mutations occur spontaneously during the course of replication. So it's potentially a huge problem, as a guy we don't have to hopefully worry about anymore. And I don't think it's going to be a miracle that suddenly one day these viruses go away. We're going to have to do something. So this talk today, I'm going to focus primarily on two proteins of interest. There's some other stories here that came out of Bill's work, but I really want to just focus on the S protein, which is the spike protein, the trimeric structure that forms the jewels, if you will, on the crown of coronaviruses. There are other proteins that are going to be mentioned in this talk today, primarily 3A. Both 3A and E are so-called viroporins. These are proteins that form channels in either infected cell membranes or potentially at least are embedded in the envelope of the virion, as well as to whether they function as some sort of a pore in the virion during any stage of its life cycle, like, for example, during the initial binding and fusion to a target cell, is really not so clear, but they do seem to play a role in the virulence of the organism, as I'll get to later. The coronaviruses really break down into a group of 4 genera: the alpha, beta, gamma, and delta coronaviruses. There are 7 of them that cause human disease. 3 of them, the really bad ones, the deadly diseases that we're familiar with, the original SARS or SARS-CoV and MERS, but now SARS-CoV-2, obviously. And there are actually common cold viruses that are beta coronaviruses, and as best we can tell, they don't cause worse disease, but they are beta coronaviruses. And then there's a couple of alpha coronaviruses that have infected humans as well. Most of these viruses that we are familiar with infect animals, and they're very diverse in their ability to infect different species. So in regard to SARS-CoV-2, most folks believe it's an evolved natural recombinant. It looks a lot like animal coronaviruses rather than more common cold viruses. Mostly it looks like bat viruses with some small parts of pangolin viruses. So it's generally thought, although not really completely proven, that this virus emerged as a zoonotic infection, probably from wet markets in Wuhan, China in 2009. At least that's where it was first reported, but there is still some controversy as to where this virus really emerged. So I'm not going to pin blame on anything, but part of the reason why we got into this story is to see if this different kind of analysis could reveal anything more about the evolution and origins of the virus. So how do we understand the emergence of human COVIDs? The RNA-based sequence alignments and mapping of SNPs are powerful techniques that certainly give you some indication of the relatedness of viruses, and generally relatedness is thought to predict origins as well. Protein alignments are useful too, primarily because amino acid changes are likely to be more important in the biology of the virus. So perhaps doing phylogenies based on amino acid sequences might be maybe a little bit more informative if it gave a result different from nucleic acid SNPs, for example. But we also were wondering whether residue divergence would influence more than just the protein that was undergoing divergence, if it would actually change interactions with other proteins that could have important consequences. And Bill's approach to this was to use amino acid residue covariance as a way of trying to map changes in amino acid residues that are linked to changes in other amino acid residues. So this is not something you think about all the time. So a couple of cartoons might help you understand what's going on here. Imagine a sequence of amino acids in a few different proteins that are related. So these could be on the left-hand side of this diagram, these could be 5 different versions of this protein from 5 different viruses. And where the red arrows are pointing to is something that could be equivalent to a salt bridge. You could think of this as a salt bridge either in the same protein, in which case it would be an intra-protein interaction, or it can be between 2 different proteins, an inter-protein interaction. Either way, just a nice salt bridge between an R and a D might explain why those residues sort of march together. But you don't really identify them as a covariant residue until you see what happens in the second line, the R changing to a D and then the D changing, in this case, to a Q. But it could have changed to a K if it was going to try to establish a salt bridge again, but maybe this is solving that problem by just generating some other kind of hydrophilic or hydrogen bond network or interaction. So the bottom line is you can think about this from a geneticist's point of view. You can think of this as sort of a allele-specific suppression. It can be intergenic or intragenic, but if you mutate a critical residue that's involved in a protein-protein interaction, if that interaction is important to the biology of the virus or the organism, you're going to need a compensatory mutation if you're going to survive the evolutionary problem that could cause. And we can think about covariance as influencing numerous things. Obviously, protein structure is where I'm starting, protein-protein interactions, but clearly they could affect the catalytic function of the protein. They could involve epitopes under immune pressure. They could involve all kinds of different mutational drifts that really might make the virus more stealthy by hiding CpG sequences and things like that. So there's lots of different ways that you could imagine covariance could impact viral fitness. So if you go to the bioarchives paper, you'll get a more detailed description of the pipeline that Bill developed. Basically, he started with 850 viral genomes. He also utilized RNA sequences to generate parallel phylogenies and protein sequences for other phylogenies. He picked the genes that are shown here, these 7 genes. You basically take the open reading frames, collect them out of all the viral genomes, line them up, do alignments, and then do covariance analysis to find out when you get a change in amino acid sequence, does something else tend to change with it across enough viral genomes that you end up with some kind of a statistical reliable signal. You can adjust these things with the software that Bill was assembling together to do this. And then eventually the residues are binned, and he uses a force mapping technique to help you visualize the data a little bit better. You can collect different categories of covariance and restrict them into different host groups. That can turn out to be something pretty interesting and functional as well. And finally, you can ask questions about the collection of covariance you got as to whether they appear in clinical samples of the virus you're most interested in, SARS-CoV-2, because that would be interesting to find out if those residues are under pressure to change or are under pressure not to change. Obviously, if they're under pressure not to change, if you can define vaccine epitopes in those areas, if that is a functionally important part of the protein, they may be more in a evolutionary bottleneck, if you will, that prevents the virus from escaping easily. What I like to use as an analogy here is HIV, that my prediction would be there'd be covariant residues that would define things that the virus can't change, like the CD4 binding site, or perhaps the CCR5 receptor, co-receptor binding site, but something like a variable loop is going to not have a lot of covariant residues in it, and subsequently neutralizing antibody that hit that loop, you can escape over and over again because it's nothing that critical. So that was kind of the baseline thought that went into this. So here's what the data set started out looking like. 850 viral genomes, 48 from bats, 5 from pangolins, 5 civets, a bunch of SARS-CoV-2s, a bunch of the original SARS-CoVs, some mouse adapted and cell line adapted mutants of the original SARS. Those genomes were available. And when you run it through Bill's pipeline, he found 866,198 covariant pairs. So you have these pairs, but sometimes they can be grouped together if the pairs are in different pairs cross each other, then they start becoming a cluster. And basically 197 total clusters were found from this database. So how do you think about this? Here's this diagram up at the top. The cluster would be all these different residues that changed in sort of lockstep with each other in multiple different genes in this example. That forms a cluster. But if you take the strains that define these changes and attach them to a cluster, you start to understand what force mapping is all about. These strains get pulled closer together if they find more and more clusters that they share, and they get pushed farther apart if they don't share clusters. So now you get to see what the data looks like. Well, it's a beautiful diagram, as it turns out, and it's really fun to look at this. And I'm going to give you the website's in the paper, but we have an interactive website where you can kind of watch this and play with it in real time. It's pretty fun. And what you can see is right away the force mapping separates these viruses quite nicely. The original SARS-CoVs are way up over here. These yellow dots you see in that cluster are the tissue culture and animal adapted versions. They don't, push those viruses out of where they belong. They are still SARS-CoV viruses. You can see that these CoV-2 strains all map together. And what is more important is most of the bat viruses are over here, but the 5 pangolin viruses and the 3 bat viruses that phylogenetically we know are very close to SARS-CoV. Indeed, by mapping covariant residues, they map physically close. And this just shows you that if you do phylogenetic mapping, either with proteins or nucleic acids, you get more or less the same result. The 3 bat viruses and these 5 pangolin viruses are very close to where SARS-CoV-2 is. So you could say it didn't change anything relative to nucleic acid or amino acid based analysis, but what it is giving you is actual data on residues and proteins, and it shows that these changes are mapping in a way that is consistent with the viruses they land in. So here's another way to look at this. This is a blow up of that region, and again, you can see all these larger dots are strains of the virus. The smaller dots, like down over here, we call these nodes, or other names we're occasionally using. They represent the actual covariant clusters. Sometimes they're a pair of amino acids. Sometimes they're a cluster of amino acids that all seem to move in lockstep with each other. So you can sort of see how the force diagram, all these different clusters are pulling things in different directions, and that's how it separates out where the strains are located. So this is the interactive website. You can get it from the paper, or you can take a screen capture or something and see it for yourself. You can query it in various ways, zoom in on clusters, ask about specific strains, search for specific genes. When you zoom in, you can find a set of strains that are getting pulled in a certain direction by cluster 180 or cluster 120, and kind of deduce a little bit more who's defining the group. And I'm going to get into that right now. So if you start saying, okay, now you got these covariant residues, they're mapping to these different strains. Can we start restricting them and mapping them in certain ways and get information out of that? And what's shown on this slide is a type of binning approach. You could, first of all, define all the covariant residues that are defined by comparing pangolin, SARS-CoV-2, and bat viruses, and then take those and say, what are the overlaps? So not all the covariant residues that SARS shares with pangolin are going to be shared by bats and vice versa. So you get this sort of Venn diagram, and then you can define the groups as either pangolin restricted. They're definitely covariant residues when you compare them to the larger database of all coronaviruses analyzed, but only some of those are shared with bats and some of those are shared with SARS-CoV, and some of them are shared with both. So you can define these different categories and then map them onto the genome. And what you see is some pretty interesting things right away. So one of the things you see is, for example, if you look at the pangolin restricted covariant residues, that's the blue triangles. Just look around the circle and you see that, wow, in between NSP1 and this spot down here, NSP4, there is a lot of density of covariation that is pangolin restricted. So that would suggest right away that that segment is likely to be the major segment that came from pangolin. You'll see other blue marks around there, but the density is obviously way higher for that relative to SARS-CoV. The the SARS-CoV exclusively restricted ones, in fact, really correspond right to that region. So that's the chunk probably that most likely came from pangolins. If you look at other restrictions like the bat restrictions, they're distributed all over the place, but they actually tell a very interesting story too that I'm going to shift to next. It's a little tricky when you think about restriction because some things are not so restricted. They're actually the category conserved A, where they're highly dense, and that's this region in here and this region in here that I'm going to be shifting to shortly. But first, let's look at the bat clusters just to get a better feeling for what's going on here. So if you look at the bat clusters, 40 selected clusters out of 48 bat genomes. On the y-axis here, we have the number of bat virus isolates that have a given cluster. So, oops, sorry. So this cluster 180, for example, has 16 covariant residues as part of its cluster. And you can see all the bat viruses have to have that cluster. They want that cluster. And then as you go down these other clusters, you start losing more and more viruses that can do without those clusters until you get to the very, very end. And way down over here where you get these only a few clusters left, those are the ones that are related to SARS-CoV. So these are the clusters that are the business end of the bat clusters, so to speak, that were maintained in the virus that we care the most about. And this is just for the case of bat viruses. So now if you go to covariant residues that were enriched, for example, these important clusters like 183, 190, 194, the ones that I just showed you, you can break them up into other groups again and go to different viral groups and map them on the genome. And right away you see something really interesting. You see that if you look at all these bat distributed clusters, the ones that were in bats that were not represented in SARS-CoV-2, for example, you see where they stack up. They stack up heavily in spike and heavily in 3A. So these are areas that had to undergo significant loss and changes in covariant residue densities in order for the SARS-CoV to emerge. And they don't match up 100% with pangolin either. So this area in here is really a very interesting area from an evolutionary standpoint. So it got us kind of focused in on 3A and spike. So here in this diagram, what Bill did was he just took all of 3A, which is this green arc over here, and all of spike, which is broken up into a few different parts. The N-terminal domain is in yellow, the receptor binding domain is in blue, and this is the sort of the rest of the spike protein, sort of the stalk, if you will, that makes it push away from the membrane. And as you can see, some really interesting things jump out right away. A lot of the covariance of 3A maps to one sort of very specific region of spike. This 16-14 proximal loop seems to be a really important area. And that area is actually tied into covariant networks in the N-terminal domain. So if you look at what's going on at 614, you find some pretty interesting things. For example, residue 614 is the location of the G614 to D mutation, which potentially makes the virion more stable and more transmissible. That is the missense mutation, if you will, relative to the early isolates that has now taken over much of the pandemic. And you find other vignettes like this that I can't get into right now. This Q23 is a really interesting cluster as well, but I can't get into it. It just takes too long. But when you do look at some of these clusters, you do see what I think is the smoking gun, what we think is the smoking gun for human adaptation. For example, cluster 126 is a set of amino acids in both SARS-CoV and SARS-CoV-2, which are all right around this Q23 mutation, which turns out to be one of these mutations that really defines the SARS and SARS-CoV lineages very, very nicely. It turns out that, as it says in the bottom, that's the only cluster of covariant residues that are present both in SARS and in SARS-CoV-2. So they may be, that end of the receptor, of the spike protein may be really critical to doing something that's interacting with something specific to humans, maybe escaping a human-specific innate immune mechanism or interacting with a human protein in some way. I know there's no evidence for co-receptors, but something more exclusively human may be going on that helps these viruses when they maintain these covariant residues. They build a structure that is doing something very important to these viruses. So another way to look at this is to map other types of functionalities onto these sites. So again, just spike and 3A here. The only thing I want to point out here is that Bill mapped putative glycosylation sites, dominant T cell epitope predictions, dominant B cell epitopes, and again, map them relative to multiple different residue variants. And make a long story short, you see these arcs are linking covariant residues to each other in various locations. And again, you see 3A is strongly interacting with the N-terminal domain of spike, not the receptor binding domain, which is down here. And the N-terminal domain is the site where you see a lot of potential glycosylations and epitopes. So maybe 3A is doing something having to do with immune evasion. And there are actually multiple monoclonal antibodies that neutralize the virus now that are directed to the N-terminal domain, and no one knows mechanistically how those work. Most people think you have to bind the receptor binding domain to neutralize this virus, but that's not true. So then finally, Bill looked at 13,000 plus SARS-CoV genomes to look for clinical mutations that arose since the virus has been in humans, and see where those map relative to some of these covariant locations. And again, to make a long story short, you definitely can see things like where conserved group A is, and some of the other clusters that we think are very important, like the cluster near 1614 DG mutation. You see that there's lots of mutations that have occurred in these clinical isolates that are linked to these covariant regions. What is not the case is that you don't find mutations in the covariant residues. This is a really important concept that I mentioned early on. That doesn't happen, and it probably doesn't happen because the virus has to mutate in two places at once to if that covariant pair is super important to something in the virus life cycle. What you do is you find mutations near those spots. So I think until we have a lot more structural biology and a lot more mechanistic work, we won't really understand what this is saying about the evolution of these protein regions and structures. But I tend to feel that if we can direct immune responses more toward the covariant residues, we might have a more universal vaccine for these viruses, especially the human adapted SARS-CoVs. So last comment about 3A. 3A is a really interesting protein that I think Vincent will like a lot because it's all about inflammation. It has the ability to activate NF-kappa B, the NLRP3 inflammasome mechanistically, not a lot of understanding of how that works. Some people think it activates the inflammasome by inducing a calcium influx, and there is some data for the previous SARS virus for that. Remember, again, all these conclusions are about SARS-1, if you will, the original SARS, not CoV-2. So as far as I know, no one's tested inflammasome activation, for example, in this protein produced by the current virus. And it's got lots of other different kinds of activities. But one of the things that is worth pointing out here up at the top is that biochemically spike interaction has already been shown for the earlier 3A and S. Not much of an understanding as to what that's all about, but trace amounts of 3A are found in virions, although it's usually not shown in the cartoons. And you can do things like pull downs and so on. So there's something going on with 3A and spike. And like I said, if you knock out either of the two viral porins, 3A or E, you can see a measurable effect on the virulence of the viruses. It turns out that either one can be knocked out, but you can't knock out both. So it looks like this virus definitely needs a viral porin, but right now it looks to be that either one will attenuate, but right now in this model, at least looking at starting weight, it looks like 3A is the more important one of the two. You get more weight loss with an E knockout than you do with a 3A knockout, and you can see over here get complete attenuation. The mice survive an E knockout. Oh, I'm sorry. Excuse me. I screwed that up. I want to get this right. So you get less weight loss with E. So E is actually the more important one for virulence in this model, followed by 3A. This is a control. I forgot. The 8A gene doesn't affect the virus virulence at all. But you can see they both matter. Okay, and then the last comment I want to make is that some of the covariant residues, for example, these two in the N-terminal domain is in the extracellular domain of 3A. Those are covariant residues that covary with spike residues. So it's fun to imagine that this domain on the outside of 3A is actually interacting with the spike trimer in some way. And then finally, SARS-CoV-2 mutations were modeled by this group. Here's the reference for it. And suffice to say that using molecular docking techniques, they found a highly statistical interaction between the 3A monomer and S. I can't evaluate these computational methods, but these guys were smart enough to take a look at the Q57H mutation in 3A, which is one of the mutations that is now becoming more penetrant in the population of viruses. And they saw a very different docking reaction for the Q57H. In fact, the docking goes up, as I recall. You have a statistically increase in the affinity of 3A for spike with that mutation, if I'm not mistaken. So you can look it up and get the details. But I think biochemistry needs to be done, and we need to understand more about the consequences of these interactions if that's what they're defining. So 3A has been looked at for antibodies. It is, in fact, a target in SARS. The earlier SARS patients have antibodies against 3A. They have antibodies against N-terminal regions of 3A, where they also find T-cell epitopes. You get CD4 and CD8 responses to 3A. There's even a DNA vaccine for mice using 3A that showed some protection. You can block 3A's potassium channel and have effects on the cell. Caspase-dependent apoptosis is altered, for example, and some ability to activate the inflammasome as well. So I think 3A is going to be a really interesting protein going forward. So in sum, then, what we've shown is covariant residues in this family of COVID viruses are skewed to certain proteins and certain domains. For example, the E protein has absolutely no covariant residues. It's just kind of wild, you know. it looked like it was the more important viral porin, but apparently there's not a lot of pressure for it to talk to other proteins of the virus. Residues S and 3A are abundantly represented as proteins that have a lot of covariant density of residues, and significant covariance is found in both the receptor binding domain and the N-terminal domain of S, despite the fact we really don't know what the N-terminal domain of S is actually doing for the virus. And there are many residues that are actually near the furin cleavage site of the S protein. So what does that mean? It could mean that 3A influences furin cleavage of S, and we know that cleavage of S is required to activate its membrane fusion conformation. It turns out furin is a protease that is regulated by monovalent cations like potassium. So one wonders if 3A isn't like somehow or another allowing monovalent cations to come out shortly after the virus binds in some way, and maybe or right after the fusion event, and then just activates a lot more furin cleavage of the initial fusion event. Anyway, lots of things to think about, but I think it's a long ways out, and it's a very early project. So I thought I might be able to do both today, but it's getting pretty late. So I was going to say, maybe we just do questions to part one. First of all, acknowledge everybody in my group, and obviously Bill for this fantastic work. We may want to save the Vibrio stuff for another day. It's a little bit of a longer story, but I think at this time, I want to thank Bill. I hear he might be online with us, and maybe suggest we start taking some questions, Vincent.
Good idea.
If there aren't a lot of questions, I'm happy to plow back in and try to cover a few more slides with the Vibrio part of the story.
Okay.
What do you think?
Let me start with one. So with the availability of the vaccine, if there's selective pressure on those epitopes, can you kind of predict what the covariant residue on for example, the 3A, and having epitope available for both of those can squash kind of the escape mutation in the future?
Yeah, that's exactly the kind of thing that we were suggesting in our paper. A bivalent vaccine with, both a 3A component and a spike component would help. Maybe you could just say that since everybody's betting their house on spike and there's going to be all these spike vaccines, maybe a standalone 3A vaccine can be used as a booster or administered at the same time, rather than necessarily asking companies to develop a real bivalent formulation in a single shot. But the logistics of vaccination is not easy. As you can see, we've been having a hard time getting even this vaccine out. So trying to ask companies to immediately work on this problem of thinking about 3A is probably going to be a difficult uphill battle. Not that it shouldn't be done, but trying to paint the virus into an evolutionary corner, if you will, I think is what one tries to do with vaccines effectively. Give the organism a very hard time of escaping the immune response, and where you have organisms that can undergo either antigenic variation or can drift their target proteins genetically or by mutational effects like flu and HIV and pneumococci makes over 20 different capsules. So I think we know that variability of the targets of these antigens are going to make vaccines fail. They may fail only partially. You may get partial protection, and that may be enough to make the disease more mild. But I think any insight we can get into improving the vaccine at this point, especially other types of interactions or conformations that these proteins might reach when interacting with other proteins of the virion, might produce different immune responses that might be able to do paint the virus into a corner.
And can you make a couple of comments about like the UK or the South African variant in terms of their covariance?
Yeah, we looked into that, and Bill can correct me if he wants to put up his hand, but we looked into it, and at first glance, both the South African variant, the UK, the South African, and the very similar Brazilian variants are not in covariant residues. I think the South African one is actually pretty close to a hotspot, though. So it may be, you know, it may be in a region that you know is like a scaffold region that it's being popped out there maybe to fool the immune response or something to go after it, but it's also escaping. I mean, that one's in the receptor binding domain, so you know it's a little more likely to be something that you know escapes the category of antibodies that hit that domain. Like I said, there's neutralizing antibodies that hit the N-terminal domain that functionally people don't really understand how that causes neutralization, but it does.
Dieter has a question about.
That's probably not a really good answer, Vincent, but it's about all I can say. Maybe Bill can comment if there's something, another point to be made there.
Hey, real quick, John, I did look at the South African variant. In the South African, and actually out of the 6 or 7, 3 of those do covary. So that one actually was one of the first covariant or first variants where we saw, you know, evidence that one change might be imposing pressure for another residue to change.
Thank you. I had forgotten that there was actually quite a few amino acid changes in that particular variant.
Yeah.
I couldn't remember the answer as to whether they were covariant or not. So that's an interesting one.
Well, it's hard to keep up with. You know, you read a paper tomorrow morning and, you know, there's so much data that shows up at the doorstep every day. So that was just from the within the last week or so. So.
Okay, cool.
Dieter commented that it would be great to use this approach to analyze or compare the host-adapted salmonella reservoirs, as well as the NTS versus the INTS strains.
That's a really great idea. I think you know the kind of the bacterial genome version of this is a work in progress, but we're optimistic that those kinds of problems are going to be tractable because the stuff we're seeing in Vibrio is certainly informative. If you know, if we didn't know more about Vibrio biology, this would have been an approach that would have told us a lot about Vibrio biology very, very quickly. So I think if we have enough genomes, I think that's the critical thing, is you need enough genomes of the 2 different types that you can say definitively, here's all these genomes that are host adapted here, and here are all these genomes that are host adapted to there. I think this kind of analysis will help tease apart which genes you should focus on for proof of concept tests as to host adaptation.
Thank you very much, John, for coming to give a great talk, and thank you, Bill, for joining us. And we appreciate it.
All right. Well, may you have another 100 great years at Eastern Pennsylvania's ASM branch. All right. Thank you for the invitation.
Yeah.